should know, the Store Lectureship was established to bring preeminent scientists to the University of Davis, and Allan certainly fulfills that. The Spradling Lab, over the past many years, has been one of the main labs that's given us a real good framework to think about stem cell biology. Stem cells, as many of you know, in adult organisms localize to discrete locations, which are called niches. Niches were first hypothesized to exist back in the late 70s, but it wasn't until a seminal paper in 2000 where the Spradling Lab described the first niche at a cellular resolution, and that was the germline stem cell niche in the Drosophila ovary. Since then, the Spradling Lab, as well as many labs of former Spradling postdocs, have gone on to further define this niche at not only the cellular resolution, but also in great detail at the molecular resolution. And it's really given us a great framework in which to understand how stem cells are regulated in adult organisms, and also really points to the value of studying this in a model organism such as Drosophila. So for his efforts, he has been awarded numerous prizes over his years. I will just give a brief synopsis of some of them. He's a member of the National Academy of Sciences since '89, member of the American Academy of Arts and Sciences, been awarded several awards from the Genetic Society of America, including the George Beadle Award, Society for Developmental Biology, the E.F. Conklin Award, and more recently, the Goober Foundation Medal in Genetics. Dr. Spradling received his PhD at MIT and then postdoced with Tony Mahowald at Indiana University, and he's been at the Carnegie since a while. He's currently the director of the developmental Department of Embryology. So, pleasure to have you.
You know you're getting to a certain point in your career when they start saying "for a while." But I am really honored to be here and very excited about giving you a flavor of the way that my lab likes to look at stem cells, just because stem cells are really a very exciting subject. They really are fundamental, I think, to the life of multicellular organisms. And if you think about a great big organism with lots of cells, even when these first appeared on the Earth, they really can't live very long based on their cells. They have to have some system of turning over cells while staying an organism, and this is what stem cells have provided. So what we're going to cover today is three basic questions. So here, first of all, in any stem cell talk, I've learned it's a good idea to define what you mean by stem cell. And here's kind of just the generic stem cell and a lineage. A stem cell, is, to me, is a cell that can self-renew. So it can divide. One of the daughters will become a stem cell, and so there's still a stem cell there, and then the other daughter will go on and do something, usually differentiate. And the major questions in understanding this powerful system are these three. One, we're going to look at how stem cells are regulated in tissues. And this is where this idea of the niche that Bruce mentioned will come into play. Secondly, we're going to look at this question of these two types of stem cells that have, over the years, been recognized. There's the very active, the easiest to study are like the intestine, the blood, the gonads that produce lots of cells all the time. They're the easiest to study, but there's other extremely important stem cells, such as those in the liver, pancreas, etc., that aren't normally dividing, but if there's an injury, they can come into play and provide crucial repair cells. Okay, so that's the second question is, are these basically the same, or are there two fundamental kinds of stem cells in these two contexts? And then the third question is one that you hear even more about in this field of stem cell biology today, which is the idea of stem cells as a source of cells of various types for replacement, repair, medical applications. And to understand how to produce, take advantage of this aspect of stem cells and how to even go from a differentiated cell back to a stem cell, what we really need to do now is step away from the stem cell and understand this lineage. What really changes as stem cells produce daughters, and they start to go down one of a variety of pathways and become differentiated cells. So what I want to show you is that by using the power of Drosophila genetics and taking advantage of the great relative simplicity of Drosophila tissues compared to mammalian tissues, we can make interesting headway on all of these aspects of stem cell biology. So let me start by a very brief introduction to the tissue that I've worked on for most of my career, which is really a beautiful tissue. It's the Drosophila ovary, which produces follicles and eventually eggs throughout the life of the animal. And these eggs, each one of these structures pictured here is a follicle that will turn into a mature egg, but it's produced from a little structure up at the tip which contains stem cells, and that's how they can do this throughout life. The structure is known in the germarium, and here's a larger version of it. And basically there's two kinds of stem cells here. One produces the inner part of the follicle, which in Drosophila consists of one oocyte and 15 nurse cells, and these cells will eventually degenerate, just leaving the oocyte. And then surrounding it are these green cells, the follicle cells, you could call them granulosa cells, and they do a lot of signaling and help the follicle differentiate. So whenever you have a tissue, you want to start studying the stem cells. The first question is, how do I find the stem cells, and how do I know I've got the right cells? And that's incredibly important, and it's been one of the problems in a lot of mammalian stem cell studies is getting close to the stem cells, but not really knowing exactly in the tissue which cell is the stem cell. And to solve this problem, there's no marker gene that will ever reliably tell you that this is a stem cell. There's no magic trick like label retention. That will never work and tell you it's a stem cell. The only way you can know you have a stem cell is to use the defining property of the stem cell, which is its lineage. It's what it's doing in the tissue. So what you need is a non-invasive way to mark the cell, just every cell, one at a time, say, and then see which ones self-renew and produce progeny, which ones don't. If they don't do that, they're going to make a clone, if you label it, what we call like a transient clone. It's just a little bit of labeled cells that'll ride along on one of these follicles and then leave the ovary, never to be seen again. But if you label a stem cell, like was seen here, and this is labeling by a recombination that generates just a marker gene that has no significance or function other than to make an easy-to-stain LAC-Z marker, then you get a clone that lasts, and it makes multiple follicles are labeled. So this is a stem cell clone for the germline stem cells and a stem cell clone for the follicle cells. And it's by making these random clones in any tissue, studying what happens over time, that you can identify, and then once you see this has got to be a stem cell clone, then you have to understand how the cells are moving and go to the head of the clone, and that will be the stem cell. So that's the basic approach to finding stem cells in all the tissues that we've worked on. And now the next thing that you want to know, let's say now you know, and if we did this, and actually if I go back here, what we found is there are exactly 2 stem cells right here for the germline, and there are exactly 2 follicle stem cells located in those 2 positions. Well, now we want to turn to this next question of what is the contribution of the stem cell to being a stem cell, and what's the contribution of the microenvironment? And as Bruce mentioned, there was this idea that microenvironment might be very important, and so this term niche was coined. But how would you know if you had a niche in a tissue? And unlike many people seem to think, like, if you had a mutant, say a mutant mouse, and it lost a bunch of stem cells, well, that means they must be in a niche, like especially if it's a signaling gene. But unfortunately, just general studies of that type, they can never really tell you whether you have a niche or what's going on. You have to get down right into at the cellular level and look at what exactly is happening by the stem cell. And in particular, what you need to do is distinguish, if you think this structure, that this little microenvironment is a niche because you see there's a stem cell in there sometimes, you basically need to be able to take that stem cell out and see if now it no longer renews. And now if you put another cell in there, crucially, then that cell can now be a stem cell. Then you know you have a niche if you can do those 2 kinds of experiments. So what we found is that the germline, both of those stem cells in the ovary actually are in niches. But the first thing that we found was kind of surprising, because people in this field had thought that stem cells are very special cells. They have stem cell genes. They're somehow the cell is responsible for this ability to maintain tissues. But what we found when we labeled a stem cell and then watched it over time is that these cells were not really that long-lived. You see, here's this decay curve of a labeled germline stem cell, and it's turning over much faster than the oocyte production turns over from that ovariole. And when we looked at the clone, so here was that stem cell clone, half, because there's two stem cells, half the follicles were labeled. The other stem cell is unlabeled and it's making the other follicles. Over time, all of the follicles in many of these ovarioles became labeled. So what we eventually realized, first we thought some of the stem cells were just being lost, but what it turns out is that stem cells can be replaced in the niche. And so in a very real sense, the niche is what's maintaining the tissue, and stem cells are just players that can play that role for a while, then be replaced by another stem cell. And they seem to be replaced by cells that are located nearby. So here's now a diagram of the stem cell niche for the germline stem cells at the tip of the ovariole. And here's a very similar stem cell niche that was characterized at the tip of the Drosophila testis. And in both these cases, we've learned a lot about the niche, what creates it, how the system works. And a lot of this work is published, and what I didn't want to spend most of my talk today is going over this published work, but to tell you about some new things. So I'm just going to very quickly summarize so you get the picture of how these niches work, is that there's some non-dividing differentiated cells, the hub or the cap cells. They have strong adherens junctions that hold the germline stem cells tightly in place, but all the other cells can move posteriorly. And secondly, they send a very local signal of BMP in Drosophila. A main ligand for this is called DPP, that represses differentiation genes. Especially there's a master differentiation gene called BAM. But it's such a short-range signal that when the cell divides, and they have systems that orient the division away from the niche, that the daughter cells get out of the range of repression, turn on the differentiation master regulator, and then lots of other genes start turning on and they differentiate. So it's an incredibly simple system based on having these differentiated niche cells that create the microenvironment, oriented division, target genes that can be repressed, and this is a sufficient system to provide lots of precursors for gametes throughout the life of the fly. Now, there are some aspects, even in this relatively simple system, there's some aspects that were not clear. And what I want to tell you now is that in addition to lineage labeling, there's another technology that's very useful, and that's live imaging. So what you have here is one of these germaria. There's the two germline stem cells. The follicle stem cells are like up there. And we've learned to incubate these and do time-lapse confocal microscopy and have it last long enough without deleterious action. You can probably watch, these cells are going to divide, these stem cells. There, one went right there, and there the other one went. Don't be too upset if you're not following every cell as we go along here. There's only about 300 cells, so this is an incredibly simple tissue by comparison to most. However, we can have software go in afterwards, and so each one of these balls represents a starting cell that then the computer followed what it did the whole time. And this marker, by the way, is what we call a protein trap. So it's a GFP that's just being expressed from a normal site in the genome, and we have hundreds of these, so you can follow different parts of the cells during these periods and see what various things are doing. This is just being used as a nuclear marker. Well, what did we learn from this? Well, one, we learned that the system is functioning normally. And the way you can tell that, if development is being maintained, the cells have to move posteriorly as the stem cells are dividing, and then everything is moving and going to a more differentiated state in the progression of development. So if you follow, there was the computer followed this group of cells. This is one of these 16-cell cysts. And over the course of 12 hours, it's basically going to become this kind of cyst. So you see it goes from here to here, and now it's one of these lens-shaped cysts. So the germ cells are progressing and developing normally for 14, 16 hours. And the question we were asking in this experiment is, what were these orange cells, these so-called escort cells? Because there's a layer of cells up here, thin somatic cells that surround the germ cells, but the follicle cells arise down here. So there's this region in between where there's only this second population of somatic cells that are interacting with the germ cells. And we weren't sure if these had their own stem cells and were moving along, or if they were stable. And that's very important to know, because to understand the niche for the follicle stem cells, we needed to know about these escort cells and what they were doing. And it turned out and you can see that from this, if you follow one of these cells, it went up to here and then it's over here, but it leaves the cyst behind. Even though these are completely wrapped by the processes of these cells, they can stay in one place and they don't need to divide as often as the germ cells. So this provided a very key insight into the anatomy of how this system works, and I think it could only have been determined with this kind of live imaging. So here we have the light blue would be one of these cells, and you see that they come up and touch the follicle stem cell. And the pink are these cysts that are moving along. So we think that there's a greater degree of stability and probably specific interactions with the follicle stem cell and these cells that create a niche for these cells, but now they have another problem. And that is that I said these cells are in a niche and can turn over and be replaced, but where would the replacement cell come from? There's no other stem cell very close by. And what we found is that, in fact, the daughters of a follicle stem cell, half the time migrate all the way over to the other side. And if there's any availability in that niche, or maybe they even push on the cell and try to push it out, but 3% of the time they take over the niche on the other side and they become the stem cell. And so you get that same result. Here's a labeled follicle stem cell. Initially, almost all the ovarials have just one labeled cell. A few have two labeled cells. But with time, the number with two goes up, because the marked one either takes over or it loses out, and then the system's stable after that. And this has led to this possibility, this interesting possibility, that stem cells compete with nearby stem cells. Like, imagine how many stem cells there are in an epithelial tissue in a mammal. And one of the problems is these cells could be, a mutant could arise in one of those stem cells. And you'd either be left with a little patch of your skin or gut or something that was very sick, or something has to happen. So what we think is that it may be that the daughters of the healthy stem cells can often just push the sick stem cell out of its niche and thus maintain a completely healthy epithelium. But this does also raise the possibility that a mutant could arise, maybe a mutant predisposing to cancer, that allows a cell to spread its genotype abnormally in favor over wild type cells. And we're looking for those types of mutations. Okay, well, now I want to turn to this question about the existence of repair stem cells, because these were well characterized. There are 5 stem cell types in these testis and ovary. But a few years ago, we started looking at the intestine, the digestive system of Drosophila, partly because the oogenesis is so dependent on digestion and having good nutrition that it's almost like it's part of the ovary, so we figured we could work on it. And one of the things we found is that the midgut, which is like the small intestine, here you see a larval gut and an adult gut. Well, the adult gut, I guess I should do this over here. It has this little region here which is acidified. It's kind of like a stomach. But this is full of stem cells, each supporting a little region of the gut. And these are very similar in many ways to mammalian stem cells. They make enterocytes and enteroendocrine cells using Notch signaling. And so that was a very nice system. But what we wanted to do is to look farther down at the hindgut, like the large intestine, because we suspected that that would be a different story and might give us an entree into repair stem cells. And part of the reason that we thought that is that, unlike the midgut, where there's stem cells arrayed along the whole length that give rise during pupal development, another great thing about working on an insect system is, you have pupation. These tissues like remodel themselves amazingly. So, if you want to talk about kind of repair and turning a tissue into something that is different but still highly functional, these tissues can do it in a very big way. And what we realized from classical literature is that the hindgut was supposed to be remodeled just from the two ends, whereas the midgut had dispersed cells, and we knew that that kind of looked like the stem cell thing. So we wondered, are there just stem cells at the two ends of the hindgut? I should point out that these midgut stem cells, this is just a little aside, unlike the germline follicle stem cell and germline stem cell, there's no evidence so far of whether this resides in a niche or not. This is like a picture of the Drosophila intestinal stem cell, a diagram, and all these cells are its daughters. And maybe we can take a clue in this case from the mouse, because recently in this crypt of the intestine in the mouse, the Cleaver's lab showed that the Paneth cells, which are daughters of the intestinal stem cell, kind of act like a niche. And the intestinal stem cells are always against the Paneth cell. They only go as far up in the crypt as Paneth cells. And so one possibility here is that these daughter cells, or some subset of them, are the niche. But this has not yet been shown whether this is the case in Drosophila. So to return to the hindgut, let me just introduce you to it in the sense that there's three classical regions. There's this region which was this anterior ring region. It's known as the pylorus. You can see it's kind of a ring, which was thought to be the source of remodeling during pupil development. And then there's the ileum is very active in water resorption and in ion balance. And then the rectum is also involved in those same functions. This was the part that was thought to come from a posterior source. And one of the reasons we were interested is there's this, if you just look with wingless, Wnt, there's a ring here in this anterior region that looked like they could be candidate stem cells. However, so let's say you want to test to see if a tissue has stem cell activity. I showed you how you could do it with clonal analysis, but a quicker way that we did it here just with BRDU and here with clones. So what you would expect if you had a stem cell, that you could label these with BRDU, and we, as our control, we're doing our midgut here with lots of stem cells. We get lots of labeling. There's only a small amount of labeling in the hindgut. And then crucially, we look to see how stable these labeled cells were. Because if you have stem cells and cells are constantly turning over, the cells that you label will go away. And that's what happens. See, in this blue bar, the midgut cells, within 3 weeks are completely gone. They actually would have been gone in 1 week. Whereas after 3 weeks, the hindgut is still almost as labeled as it started out. Secondly, if you really had anterior stem cells that are moving posteriorly, if you just calculate the average position of labeled cells, you should see a movement over time. But we saw no movement posteriorly. And then we also looked for clonal induction. You should be able to induce stem cell clones, but you can't get any clones above background in the hindgut. They're stable, et cetera. So it turned out that previous claims that this tissue was maintained by stem cells were due to the background induction, as we showed here, background induction of embryonic clones during adulthood, just spontaneous induction. However, what we did find, normally this is a very stable tissue. It doesn't need stem cells to constantly replenish its, maybe it's not as stressful or something like that, as in the midgut where there's bacteria and toxic chemicals, et cetera. What we found, however, is if we damage this, and we did this by expressing GAL4 and then a UAS apoptosis-inducing genes, and then we also have a repressor, so it only turns on when we raise the temperature, we could get lots of these cells to start dying. And when we did that, we saw a burst of BRDU incorporation. As you can see, there's very little without raising the temperature. And also phosphohistone H3, and it's all around the wingless ring. So there starts to be a lot of activity, a lot of cell division, but we wanted to determine if that was the damaged cells were just like repairing themselves, or is this an induction of some kind of cell that can actually produce daughters that can recover from this tissue damage. So what we did is what's called a flip-out experiment, where we only activate the apoptosis in a few cells where this stop cassette is flipped out of the GAL4, and that also, those are marked with pink. So the pink cells are undergoing apoptosis, but the BRDU is in lots of other cells that are not undergoing apoptosis, showing that it's kind of acting in trans. It's the presence of damage is inducing activity. And then we also could show that these cells, unlike the control, these cells do spread down the pylorus from this region. So what we think is that there's that this ring of wingless cells, either the wingless cells themselves or nearby cells, are quiescent stem cells that can be activated by local damage. And now we have in Drosophila a system where we can study quiescent stem cells as well as what previously has been studied, which are highly active stem cells. And we can see if repair stem cells also have niches and whether they also respond to the similar kinds of mechanisms and signals that run these highly active stem cells. And I should just at this point point out that actually the escort cells seem more like these inducible. They're kind of partway in between maybe, but I showed you before that they don't have to divide to keep up with every germ cell that moves along because they can kind of pass these off to their neighbors. But if there is a change in the number of germ cells, for example, by just artificially by mutating, upregulating some of the signals that affect the niche, or mutating if some of the germ cells don't differentiate properly and they keep dividing, like in a tumor, these cells will start dividing and continue to ensheath all the germ cells that are produced. So they have the ability to recognize a certain situation, in this case, the proliferation of excess cells, and to divide in response. So they're like a kind of quiescent stem cell as well. Okay, I want to take a slight detour now and tell you about something that's maybe it's not exactly stem cell biology, but it's really of interest in oncology, and it's also part of differentiation, and that is the production of a polyploid cell. So in mammals, it's very well known that especially endodermal tissues like the liver have a lot of polyploid cells. And in Drosophila, this is even more common. Differentiation usually consists of a certain number of mitotic divisions and then a couple endocycles. And generally, these cells, they're undergoing an endocycle. They just get bigger, but they don't divide. And yet it's been shown that a lot of cancers have polyploid cells. And it's been proposed, and there's experimental support, that some of the genomic instability in cancer comes from like tetraploidization, and then that creates much less stable division, so then chromosomes start getting lost. So, but there hasn't been any system in any organism where there's normally a polyploid mitotic cycle that I'm aware of. However, here we were working on the hindgut, and in the 30s, there were claims that in the mosquito, that the hindgut cells in pupal development underwent mitotic divisions. So we thought, we should really look at this. We're getting kind of used to looking at hindguts. And in collaboration with Joe Gall, this, for example, first this is from these old papers, and they show mosquitoes normally have 3 chromosomes, and here you see 3 bundles of chromosomes in prophase, and then they go onto a metaphase plate, and that's like a 16N cell, but it's dividing. So we wanted to see, in collaboration with Joe Gall, a postdoc in my lab, Don Fox, we got some mosquitoes. We looked at their pupae. We found the time. They said it would start at 8 hours. The papers did. We looked at 8 hours. Sure enough, you started to see these mitotic divisions in this hindgut, in the ileum part that I was telling you about. And it's only a certain period of time when the number of cells is increasing. But as you can see, there's not that hard to find these things. And so we calculated how much these ileal cells, these polyploid cells increase. And then we addressed this issue of are they just making more polyploid cells, or are they actually changing their level of ploidy? And the levels of ploidy on average seemed about the same before and after. So what it seemed like is it was just a way for cells that were already part of a tissue and differentiated in a sense and polyploid to just make more cells. And we really wish there was a way to study this in Drosophila where we could get at the genetics. And we looked at the ileum in Drosophila, and it just turns over during pupal development. So that was not a source of similar cells. But we were very surprised to find that there was a region of the gut in the rectum where we saw very similar looking mitotic divisions, and this is a lot more chromosomes than Drosophila normally has. It was also very precise, and it also involved an increase in the number of cells, about a 4-fold increase. And you get nice polyploid mitoses. You see the somatic pairing, like they're the 8 fourth chromosomes, for example. So where was this? Well, to return you to the hindgut, here's the pylorus, the ileum, and then there's this rectum in the adult. And we kind of reanalyzed this old view that there were these 2 rings was far too oversimplified. But what it turned out is that this whole pylorus, actually, the anterior part makes the pylorus of the adult, the posterior part makes the ileum, and then the rectum comes from 2 sources. One was from this A10 segment of the genital disc, and then we found that the rectal papillae cells come from the old larval rectum, which was thought to just turn over. So what are these rectal papillae cells? Well, they're like tubes that go into the rectum just to give more surface area. And so they do even more water pumping and ion balance. And like in a saltwater species, these things become really elaborate and provide the ability to tolerate salty environments. But what we suspected is that these 4 pieces come from the old rectum of the larva. And here they have very interesting outer parts, which are labeled here. And then here's just some markers that label these cells. Here, this graph shows that they are polyploid, 8 and 16C. And these cells seem to behave a lot like the ileum in that here they are, they break into 4 pieces, cells from the genital disc surround them, and then they increase their number 4-fold by going through a couple rounds of polyploid mitosis. And when we looked at the cell cycle of these mitotic rectal papillar cells during this key period, we saw all the normal characteristics of a cell cycle. They had early S, late S, fluctuating cyclin E and cyclin A. They have a nice cytokinesis, anilin in the ring, in the EM, the rings look normal, the contractile rings. But that still leaves the question of maybe they were just almost finishing. So what we did is live imaging, and we could just follow these cells going through and completely separating into 2 cells. And here I have one of these. And again, you see just the high degree of synchrony, because there's just cells like going into mitosis all over the place here. This is a marker called Jupiter, GFP. It's a marker for a cytokinesis protein. So the remnants of the cytokinesis stay around a little while before they dissolve. So that seems to be a normal cell cycle. But this is kind of raises some problems. Because the cell cycle in Drosophila, in endocycles generally, here you have the normal part of the cell cycle, and it's protected so that replication is complete by checkpoints. So if there's a DNA damage checkpoint, if there's still an ongoing replication fork, that will inhibit the entry into M phase. But in an endocycle, the genes of M phase are turned off. So there's no replication checkpoint to protect that cell from turning off its cyclins prematurely and then just resetting and starting another cell cycle, which might leave part of the genome unreplicated. And the problem with that is then if you want to go into a real mitotic cycle later, you could just rip the chromosomes apart because there's a part of it that's not replicated, and there's no guarantee, there's no way for the cell to know that. And this is probably most dramatically seen in the palatine chromosomes of the salivary gland where the cycles are not complete. And so a lot of the late S replicating DNA, repetitive DNA of the genome, is completely underrepresented. It goes down hundreds of fold because it's just not replicated in these endocycles. But these wouldn't contain the centromere region. So how could a cell that had gone through cycles like that ever have functional centromeres and divide? Well, what we found is that although the ileal cells undergo salivary gland-like cell cycles and would not be, have normal centromeric DNA, the L2 cells, way back in L2, the cells that are going to become these rectal papillar cells have normal cells. So they have a different cell cycle, almost like an anticipation of the fact that they're going to become mitotic. And this actually, a similar state occurs in the nurse cells, where for the first cycles of the nurse cells, the first 5 cycles, they go through complete replication. Then they start to go into M phase and the cells fall apart, and this creates a situation ultimately where you have polyploid polyteny, but they never actually make it through a cell cycle. So we were very interested and surprised to find a cell that does this complete replication and then actually goes back into a regular mitotic cycle. And we were able to find some of and test the fact that regular cell cycle genes are important in this. We also found that Notch is important in inducing and determining the number of rectal papillae. And if you express a gene called Fizzy-related, which is part of the APC complex and is needed to enter endocycles, but it has to cycle. So if you express it constitutively, further divisions are blocked. And these things all fail to undergo the 2 divisions and just stay with 25% of the normal cells. And the same is true if another gene string is mutant, you can't go through, it's not a completely tight mutant, you can go through 1 but not 2 divisions. So that brings me to the final issue with these cells. Now that we have a natural in vivo cell, a polyploid cell that's undergoing divisions, are they error prone? Like that would be argued from these studies. And remarkably, we see lots of chromosomal mistakes in these cells. So just looking either in both mosquito and in Drosophila, in these cells, we see a significant, like 15-20% of the mitoses have a lagging chromosome. So for some reason, even though this is an error-prone process, these cells are using this to generate this last 2 divisions. And it provides us with a very nice system for studying the genetic requirements and consequences of mitotic polyploidy. And why might this be? Well, recently it was shown that polyploid cells, in at least some of these endocycles, don't even use ORCs, that they replicate their genome using a different process. They use a lot of the downstream machinery, but they may actually recognize origins with a different protein and only use ORC6 to bind to CDT1 and CDC6. But that could mean that there's a less perfection in terms of ensuring uniform replication, enough pre-RCs to not leave any regions unreplicated. And another thing, Brian Calvi's lab showed that polyploid cells, when there is damage, don't go into apoptosis. So the combination, so in other words, these cells are naturally like tolerant of DNA damage and may have a damage-prone system. This could explain why there's enough damage there that you get these lagging chromosomes. Okay, well, I want to turn now to the third question that I raised initially, which has to do with stem cell differentiation. And in particular to this question of epigenetic inheritance, because it's epigenetic inheritance, meaning inheritance of a trait throughout cell division that's not encoded in the DNA. And that's a key concept and a key process for multicellular differentiation. You're starting with a stem cell that can become multiple types of cells, and the reason it becomes one type and not another type is most likely involves, at least in part, epigenetic inheritance. In principle, it could be that there's just signals over here that make it turn on all the genes to be that type, and if you took it out of that location, it would no longer be that cell type. But we know from transplantation that doesn't seem to be the case, and that there's heritable changes going on. So how can we study these and identify what are the changes and what are the mechanisms of these changes? And I think stem cell lineages are extremely valuable for this. Just like you need lineage analysis to find stem cells, you really need lineage to study epigenetic inheritance. And part of the reason is if you're looking at a gene, this light green gene symbolized here, and you see a change, if you can't follow the lineage, you'll never know if it's epigenetic because you have to show that it's, whatever that change was inherited. Then you know it was an epigenetic change. So you have to follow lineages to know if changes that you're seeing are epigenetic or just fluctuations. Now, even if you know that you've got some change that's epigenetically inherited, there's still two major types of epigenetic inheritance, you might say, that often are not distinguished in the field. There's what we all kind of wish was, epigenetic inheritance, and that is the inheritance of non-DNA sequence during DNA replication. So this is like a protein complex, a secondary modification. The DNA replication fork moves through this region, and you get epigenetic inheritance of this complex if somehow now both strands have the same complex at the same place. So whatever information or significance that had for the cell, both daughters will have that same information, that same significance. But this has actually been very difficult to study, and there's not really any known mechanisms except for like methylation, where there's a maintenance methylase. But for protein complexes, there's no known mechanism for ensuring that the two strands have the same proteins in the same place. And the other thing that can look just as much like epigenetic inheritance is diagrammed here, and that's where you have gene networks that feed back on their own production. So like if you turn on a gene that activates itself, and then the cell divides, the cytoplasm will contain lots of this protein. It'll activate itself in both the daughters. It'll look like epigenetic inheritance, but nothing's happened on the chromatin that is like what we're thinking about. So what I want to tell you about is a way to look at epigenetic inheritance in a stem cell lineage that lets you find this type of epigenetic inheritance and study it in detail. Okay, so how can we do this? And what we want to do is turn to a stem cell system that has a long lineage with lots of cells to look at. And that's the follicles. The follicle cells of the ovary are premier in this case. They have 9 mitotic divisions and then 3 endocycles. So these 2 stem cells, which I told you about before, each one only divides once to make all of the 600 and some follicle cells over a follicle. And another absolutely needed property that these cells have beautifully is that they have to divide in a very simple, uniform way. So here you see 3 genetically induced clones in the follicle cells. You see each one is a little cluster of cells. They're not exactly regular, but they were induced at the same time. They all have about the same number of cells because these cells all divided about the same rate, and they don't migrate away from each other, even though they slide around a little bit to make these rough edges. So if you saw a cluster like that, you could say, if there's 16 cells, well, that happened 1, 2, 3, 4. That happened at division 5. That was just one cell. Something happened. And we know in this case it was an FRT-mediated recombination. But if it was epigenetics, you could say something happened epigenetically right at the division, just from the characteristic of the clone. Okay, well, where do we get these kind of epigenetic variants? Well, what we realized is that GAL4 UAS genes provide a huge wealth of epigenetic variants to study in Drosophila. And so here you see the GAL4 UAS EGFP-driven pattern in the follicle cells of a typical pair of constructs. And these are used like all the time in Drosophila genetics and in many other organisms. But you see, this is not like one uniform green color. It's patchy, and these patches reminded us very much of these clones. So here, for example, there's 4 cells. So we imagined that 2 divisions ago, there was just one cell that upregulated its programming of this gene, and then it was inherited. That new programming, whatever caused it, was inherited. Or here's one that there's 2 cells that are off. So 1 division before the end, something happened, the programming of this gene was turned off, and it was inherited to be off, if this interpretation is right. And it's important to notice that some of these clones are big, and they actually, if they're clones, then the events that they would represent are happening well before GAL4 is ever produced. So it's some kind of programming that is manifested when GAL4 is produced, but it's not the level of GAL4 necessarily. Okay, well, first most people looking at this who know Drosophila would just say, this is position effect variegation. So I don't know how many of you are familiar with this, but if you take a gene, that's in a normal part, euchromatic part of the chromosome, and move it to a region of heterochromatin with lots of repetitive DNA, it doesn't just turn down, but it becomes unstable, and so you get patchy expression. What it means is that doing this affects cell lineages differently, but it's never been understood what that patchiness is or why that happens. But we wondered if, in other words, this was gene silencing. But we'd seen these patterns in 4 different GAL4s driving 3 different UASs and all combinations. So, and we looked at where these were. None of them were in heterochromatin. So it didn't seem to be a heterochromatin connection. And next what we did then is we decided we would test whether this was gene silencing by using trans modifiers. So one of the great things about position effect variegation, and here's the classic white modeled 4, where the white gene variegates in the eye, and then you can find these genes on other chromosomes that dominantly modify, in this case suppress the variegation, so you get full pigmentation. This gene, SUVAR39, turned out to be histone H3K9 methylase, a gene involved in making heterochromatin. This is how it was discovered. This is how lots of genes involved in silencing were discovered. It's a very powerful system. So we got a true insert in heterochromatin. It expresses in only 50% of the follicle cells, and we showed it responded just like you would expect to every modifier we tested. And then we did our GAL4 lines, and they didn't respond to anything. They didn't respond to SUVAR39, as shown here. They didn't respond to extra heterochromatin, to temperature, to other modifiers, and none of the genes were in heterochromatin anyway. So we think that this variegation here is not gene silencing. It also didn't have to do with the tandem repeat of the UAS, which that showed. So let me just show you that we could prove, however, that it's true epigenetic inheritance. And so the way we did that is in a GAL4 line, we generated real clones, and then we compared the genetic clones to the expression. And if it really is epigenetic, they have to correspond because, it would be a change, and then the change would be inherited. So to see that, if you just look here, here's a rather large clone we induced with the 19 some or 21 cells, and they're marked with red nuclei, and they kind of cover up the expression. So if you look over here, the same clone is outlined with these little yellow balls, so you can see the expression pattern within that clone. And what you'll see is that there's a perfect correspondence of the clone to one of these zones of expression. And if you try to fit that clone into any other part of the follicle and have it correspond, it's impossible. There's no other way that clone of that shape can fit on here and be in one expression zone. And that's when we started going through, and that's what we found again and again and again. The only other pattern we saw was that sometimes there was a secondary change wholly within the clone, because as it was growing, there was a second change. But then that secondary change was totally within the clone, so that also fits the rule. And this proved to extremely high uncertainty that these clones are caused by epigenetic inheritance. We could also look at a part of the pattern where there's a big clone and see a little clone that's totally inside of it. And remember, there's not much cell movement. So that means that the little late clone came from the surrounding tissue. And that would let us ask how many times does the expression go up and how many times does it go down? And we found is it's 50-50. So this also doesn't fit the gene silencing idea. And we also could look for twin spots, as if it's something that's limiting being lost. When one cell goes up, does its other daughter go down? That would makes a twin spot. But we did not see twin spots. This level, where it looks like a twin spot, is probably just coincident clones, and the others, there's just one, not two. Okay, so now what I want to do is take advantage. Now we've got a system. We can look at epigenetic instability. We can map when it happens. So we wanted to see is during stem cell differentiation, is epigenetic inheritance equally stable the whole time, or does something change? And so to do that, and you can actually just tell at a glance here, but we just circled all the clones of various sizes and totaled them up, and here's that for 6 different lines. And then for each one of these, like say the number of 2-cell clones, then we calculated how many 2-cell clone divisions actually are there in this number of follicles. because you want to know both the number of times something changes and how many times it could have changed. And there's a lot more late divisions than early divisions. And what you find is that there's a tremendous change in epigenetic instability during differentiation. It's quite high. 15% at division 4 goes down to like 0.3% at the last division. And this is true in all the lines. You get the same curve. Well, we wanted to go all the way back to the stem cell. And so to do that, we couldn't extrapolate that many divisions back on these late follicles because the clones are just too big and too many secondary changes. But we could go to the earliest follicles, use 2-photon microscopy, map using software the little clones on these, and then we could get the last divisions, which overlapped to make our composite stability curve. And we could go all the way back to the daughter of the stem cell. The stem cell itself only contributes one cell to each follicle, so you can't look to see whether there's any instability in one cell. But the daughter, which can replace the stem cell and become a stem cell, and we think is at the same level of differentiation, 40% of the time it changes its programming. So it's very, very unstable. And then it goes to where there's almost no changes, 0.3%. So there's a tremendous increase in stability and a tremendous increase in the ability to inherit epigenetic information during follicle cell differentiation. In fact, this actually suggested a model to us that stem cells and pluripotency or multipotency might be the state of not having epigenetic inheritance turned on. Because when you think about it, it would be a really great mechanism for keeping a cell from differentiating. Because if it got a sporadic fluctuation in gene expression, a little, signal, a little notch signal comes in from one side or something, and it starts to think I should differentiate, it cannot transmit that information to any daughters. It can't. It just can't even remember that it was supposed to do that. So I think that keeping stem cells, epigenetically naive, and then only as they differentiate, turning on the system might be very fundamental. It's certainly also this curve can explain why, well, if going back to the cloning, the earliest cloning experiments in Briggs and King, they saw this tremendous difference. When they took blastulin nuclei and put them into an egg, it could develop normally, very frequently. But when they did nuclear transplant with later nuclei, even not that much later, they were very, very rare that you would get development. And this same phenomenon has been seen throughout the whole history of cloning by nuclear transfer. Now it's being all repeated with iPS cell experiments where, if you're early up on a lineage, iPS works a lot faster, a lot more frequently than if you are taking from a very differentiated cell. And so these differences in internal instability could be the reason. It's a lot easier to reprogram something if they're fluctuating anyway than if they're very stable. But I promised you I would say, I would address this question of whether, is this really the kind of epigenetic instability that involves chromatin, or is it just more gene networks doing their thing? So to address this, we looked at a GAL4 that has two different UASs. The UASs are the same, but the marker gene is different. Okay, well that lets you detect whether the variation is a trans factor like GAL4 or any factor, could be RNA polymerase or anything in the cell, or is it a cis factor? Because only if it's a cis factor can these things behave separately. Like one of them could change its expression, the other one in the same cell, the same lineage of cells, not change. And so that would mean you either have yellow clones or you'll have red and green clones. And what we see is we get both kinds, but we get lots of nice red and green clones. And those have to be cis-acting, and that argues that we're seeing true cis-acting epigenetic inheritance probably from the chromosomes, specific locations on the chromosomes, and that's where the instability is coming from. And just mapping the cis-acting, you see this same tremendous stabilization during differentiation. So we don't have to, we can look at our yellow clones, but the important thing is we have lots of these clones to study. And it's interesting to see, why don't genes all look variegated? And what we think is that the times we're talking about are rather early, and here's the program of follicle differentiation. But the mitotic divisions of the follicle cells are rather early. You know, they start here, and you're already at division 5 by the time you have a follicle, and then you just go a few more, and now you're starting to get very stable, and then all the interesting stuff starts happening. In fact, if you read papers on this system, most of the papers, they don't even pay any attention to this. But they're looking at all these interesting stages, and there's signals, there's AP axis, you know, DV axis. So that is not based on lineage. None of it has anything to do with lineage, as you can see with this gene expression profile. So the lineage is not really that useful for making a functional structure. It doesn't, you know, it doesn't have symmetry. So what we think is that the fluctuations and the changes in gene expression are an early phenomenon, but they're overridden by signals to produce the beautiful final patterns of the egg. Yet they're, during those early times, they may be quite important, and they're also an indication of the changes within the machinery that's going on during differentiation, which is very important. If you want to change that machinery, if you want to reprogram a cell, this is exactly the kind of machinery you want to know about so you can change it or reverse it. Well, so I haven't told you, what I haven't told you is this is a great genetic system, but what is changing? Like, what is it that's unstable and becoming less stable? Well, we don't, at the moment, we don't know for sure, but there are some interesting clues. And one of them came from looking at the cell cycle of these cells during differentiation. Okay, so this is just a BRDU pulse. And here the stem cell region up here, and you see that very few of the cells in this 1 hour pulse got labeled and turned pink. But later, a bigger fraction, and then more and more of the cells are in S phase as differentiation proceeds. So in other words, S phase is getting longer as part of differentiation. And this is kind of generally true in embryonic development. Early embryonic divisions are often very fast, and then they get slower. And another thing that changes is the pattern of DNA replication. So the reason they're so fast is they have a lot of origins, they're randomly selected, and later sites of replication start to get much more specific and more related to the ultimate pattern of gene expression within the cell. So this, to us, seems very much like what we're seeing. We're seeing a lot of fluctuation, like the random origin selection early, and then a much more fixed pattern later. So what we could imagine is that we're putting one of these markers into the genome, and if you know that where an origin relative to a gene seems to affect how highly it's expressed. If the gene is if the origin's right near the 5' end and it's a very early S gene, that's what you see for highly expressed genes. No one really knows the cause and effect that well. And like if you have an origin that's a long time away and the gene replicates very late, that's associated with a low level of expression. So when we have these random inserts, you know, and first the genome is there's a replication in this cell here, but then later it turns and starts replicating somewhere else. Well, that could be why we're sometimes seeing a higher level, a lower level, why it goes up or down, and then ultimately the pattern of replication gets fixed, and whether you're in a good location or a bad location gets fixed. You know, and maybe then these effects of this can be overridden later in development, but we think that what we have is maybe a marker for the evolving patterns of replication in the genome and how they relate to the sites and the genes at those sites where we're looking. So this is just to summarize that stem cells are in niches and this actually suggests if a stem cell doesn't have much ability it just listens to the environment, it doesn't have any memory in its own chromosomes, that could explain why it could just sit in a niche and the niche can tell it what to do. And it's only after it leaves, you know, and it starts to gain the ability to respond and remember, it can go through a program of differentiation. So how are we going to test this? Well, ultimately, you know, I gave you a model with this replication, but it doesn't really matter what we hypothesize, because with Drosophila genetics, we have a process that's going to lead us to the answer, completely independent of whether we guess the right answer or not. Because this is just like position effect variegation. We can just find modifier genes, ones that make the variegation be stronger or weaker, and then clone those genes, and we'll find out what the genes are that affect this process that's distinct from PEV modifiers. And we found that there are modifiers. In a few years ago, which really got us interested in this variegation, we found that a gene called poly-ADP ribose polymerase, the most abundant nuclear enzyme, which puts ADP ribose on histones and is a very interesting enzyme. It's responsible for puffs in the polytene chromosomes, is a very strong enhancer of GAL4 variegation. So what we're doing now is we're starting to do screens for modifiers, and we think that they will point to machinery such as these green proteins that I have diagrammed here, whose job is actually to make sure that complexes and modifications following DNA replication are equal on the two strands. Otherwise, they put a checkpoint in, it slows down until they become equal. That there's actually a chromatin, like chromatin polymerase, that you can't just, chromatin won't just duplicate itself on its own. It's going to need machinery to do that. And we think we can find it using stem cells. And I just want to give credit to the people who did the work I talked about. Don Fox did all the work on the hindgut, and the mosquito part was in collaboration with Joe Gall. Lucy Morris developed the live imaging technology, used it to study escort cells, and did a lot of other work I didn't have time to talk about. And Andrew Scora, a grad student in the lab, developed the follicle cell epigenetic system. And thank you very much.
So have you looked at some epigenetic chromatin marks in stem cells, and are they underrepresented relative to more differentiated cells?
We have gone through with some stains, antibodies for various modifications of histones, but and we see various things. Sometimes they're strong, sometimes they're weaker. I don't think we can say for sure. Those are aspects of chromatin, but whether they're really epigenetic is not so clear to me. Because in most studies, it's not clear whether high levels of these histone markers are an effect of gene transcription or a cause of gene transcription. So, I think that this is a way we can test, perhaps, at least for this aspect of epigenetics, the role of some of those processes. But it's not like these cells aren't transcribing anything, and so we just don't see nothing that simple pops out, or it probably would have been found, before this. Yes?
Do you imagine that this having lots of replication forks is somehow equivalent to diluting out the chromatin inheritance machinery?
No, it's if you have lots of replication forks and they're thought to be induced randomly, no one really knows how you go from random to non-random with replication. Okay, but it is known it starts out, there's just a huge concentration of replication factors that just force themselves onto any AT-rich of the part of the genome, and that's why you can have a 10-minute S phase and replicate the entire Drosophila genome during cleavage phase. And so what that means is that any gene, most genes aren't even on in that situation, but that means that in some times a gene will be replicated in this favorable way by a polymerase passing in the right direction nearby early in S, and sometimes it'll be replicated much later because there was only a polymerase, there was only an origin a long way away. And the reason that sometimes can affect a gene is if the level of a key transcription factor, it's thought that replication forks in some cases knock transcription complexes off of the chromosome. They have to be reassembled. Maybe there's a partial mark of that. But then if you're late in S, other promoters have grabbed up a lot of these factors, and so there's not very many of them left. And so that gene won't be transcribed very efficiently. So that's one of the thoughts on why time in S of replication can affect a gene's transcription level. So all I was trying to say there is that the characteristics of replication fit what's varying here. Yes?
So I'm curious as to the pictures that you had of the polytene chromosomes and the breakage that you saw there. And I was kind of reminded of just curious as to what you think the mechanism is of the breakage, and if you've looked at to see if neocentromeres might have been formed because of all the the replication. Anyway, just what do you think the mechanism might be, and if you've looked at
Well, the mechanism, the reason that there's thought to be a lot of breakage during an endocycle which doesn't complete S phase is there's still forks that are unfinished. But when you get to the end of S phase and there's a fork, most studies that have been looked at that is the fork breaks. It doesn't stay there like through M phase. So that's converted to double-strand breaks, and then they're repaired by double-strand break repair. But double-strand break repair is not always perfect. So you can, if, for example, it was repaired to a different chromosome, now you've got a translocation. And then if that tries to go in mitosis, you get all the same problems you get from other chromosome rearrangements.
So it looks like you still had, that there was some, there was some, do you think that there was a chromatid bridge, or do you think that's a protein bridge that you see there?
No, those were like chromosome bridges. Like the chromosomes are being pulled in a way that they can't separate. They're both attached to the same, the same chromosome is attached to both sides of the spindle or something like that. So those, they're lagging, and then they may break, or they may just be lost when the nuclear membranes reform on the two daughter cells. But remember, they're polyploid. So this is not as serious as if you're a diploid cell. Remember, the cancer cells have lots of changes in their chromosome. They're pretty happy cells. So the more polyploid you get, the less you care about really accurate segregation of chromosomes. In fact, it could generate useful variation. So, maybe there's doing this because the rectal cells micro-adapt to certain, regions, and this helps them be selected, or they move around. Like, I don't know. But this is what we see happening. It's in a normal fly, this is what goes on, and so now we can study it and learn how this works, and then eventually maybe we'll understand why they would want to do that. Yes?
I have a question about your quiescent stem cells in the hindgut. I noticed in the picture that it seemed like the quiescent stem cells were often neighboring the apoptotic cells. And my question is, do you think that they're true stem cells, dedifferentiated or undifferentiated stem cells that produce daughter cells that self-renew and differentiate cells, or are they just neighboring cells that have lost contact inhibition or are dividing to fill the void?
Well, that's a really good question. And I hope, I don't consider us to have proved that those are stem cells. I mean, I certainly in our